Bioremediation is a process that uses mainly microorganisms, plants, or microbial and plant enzymes to detoxify contaminants in the soil and other environments. With an exponential growth of human population, the amount of contaminants in soil, water, subsurface material, and other media have been simultaneously increasing. For example, according to World Health Organization, globally at least 2 billion people use a drinking water source contaminated with feces. As a result, more than 800,000 people are estimated to die each year from diarrhea as a result of unsafe drinking water. One of the solutions for the problem is bioremediation. Currently, there are 3 main types of bioremediation, which are microbial bioremediation, phytoremediation, and mycoremediation. Microbial bioremediation uses microorganisms to break down contaminants by using them as a food source, while phytoremediation uses plants to bind, extract, and clean up pollutants such as pesticides, petroleum hydrocarbons, metals, and chlorinated solvents. And the third type, mycoremediation, uses fungus digestive enzymes to break down contaminants. But how does it work? Most bioremediation processes involve oxidation-reduction reactions where either an electron acceptor is added to stimulate oxidation of a reduced pollutant, or an electron donor is added to reduce oxidation pollutants. In both of these approaches, additional nutrients, vitamins, minerals, and pH buffers may be added to optimize conditions for the microorganisms.
So bioremediation techniques basically divides into 2 parts. First, ex situ. It's about land farming, biopiles, bioreactors, and composting. Second is in situ. It's about natural attenuation, bioventing, bioslurping, and biosparging. So, in situ bioremediation is the application of a biological treatment to clean up hazardous compounds present in the environment. The optimization and control of microbial transformation of organic contaminants requires the integration of many scientific and engineering disciplines. Some of in situ bioremediation practices will be discussed below. So first is intrinsic bioremediation. Biostimulation involves the modification of the environment to stimulate existing bacteria capable of bioremediation. This can be done by addition of various forms of rate-limiting nutrients and electron acceptors, such as phosphorus, nitrogen, oxygen, or carbon. Example could be ZOB1 was used as a biostimulator and biofertilizer for rice crops in Kozlorda province. Another branch is engineered in situ bioremediation is biosparging. This involves the injection of air under pressure below the water table to increase groundwater oxygen concentrations and to enhance the rate of biological degradation of contaminants by naturally occurring bacteria. Biosparging increases the mixing in the saturated zone and thereby increases the contact between soil and groundwater. The ease and low cost of installing small diameter air injection points allow considerable flexibility in the design and construction of the system. So the example could be widely used treat contaminated soil of oil-producing territories in western Kazakhstan. While ex situ bioremediation describes a process where contaminated soil or water is removed from the environment by biological organisms. Ex situ bioremediation can use bioreactors and added nutrients to speed up the breakdown of environmental pollutants. Some of the ex situ bioremediation practices are followed below.